Hey guys, so back again. Um, as always, let's talk about what we're doing today. So, you are going to do that Hardy Weinberg FRQ practice question that we didn't get to last time. Okay, so you're going to have some time to do it in class and you're going to turn it in. Then we're going to do a quick little recap on phylogenies and phylogenetic trees, and then we'll do a FRQ practice over phylogeny. This video should be much shorter than last class's video because once you are done with the things that I'm going to go over in the video, you will then spend some time working on another phylogenies practice. It's a packet. And then you're going to work on some types of selections and a little bit of graphing. Okay. Tonight's video, I believe, is speciation. So make sure you grab the note sheet that goes with that. In terms of our focus, we're going to continue to practice breaking down free response questions so that you are comfortable with them and you understand what they're asking you to do, and you have all those little strategies and tips and tricks that we've been talking about all year. Or we'll continue to talk about, I guess, all year. We're going to review some of the more important phylogenetic terms. You did already have notes over this, so you should have your notes with you. Again, the goal is not to learn it all from the video. The goal is to be exposed to it prior to coming to class. Then we're going to practice drawing and labeling a proper phylogenetic tree and being able to justify why we place the organisms where we place them on the tree once it has been drawn. Okay, so at this point, you might want some note paper so that you can kind of write down what I write down, or if you have space on your notes, or however you want to do it. But I'll give you a minute or two to orient yourselves. Okay, so hopefully you have your paper and you're ready to go. So phylogeny is the study of evolutionary relationships. This should sound familiar from last night or the night before's video. And by evolutionary relationships, we're pretty much talking about relatedness. How related are you to another species, another individual, whatever? So a phylogenetic tree is just a diagram that gives an image to these relationships. Okay, so it's a diagram that represents evolutionary relationships. Okay, so the branching and the tree itself reflects or shows how different species or groups all evolved from what's known as a series of common ancestors. Okay, so you and I are related because we have this person in common. You and this other person is related because you have that person in common. Think of it from that perspective. Okay, so we're going to say that the branching pattern shows how species or groups evolved from a series of common ancestors. Okay, so some things to remember to keep in mind is the more related two groups are, the more recent their common ancestor. The less related they are, the less recent the common ancestor will be. Okay, and then another thing to keep in mind are the branching points are mobile. So if you rotate them, if you flip them around, you don't actually change the information that that tree is giving you. So there are lots of ways to draw a tree, in other words. Okay, so let's talk about what the tree looks like. Okay, so remember that your phylogenetic tree is a hypothesis. Okay, it's your best guess, it's your best estimation at what a series of groups would look like based on common ancestry. Okay, so it's gonna it may look different depending on who's drawing it, but because it's a hypothesis, that's okay. Now, within reason, it has to look a certain way. We have to have some sort of order for it to be considered correct. But at the end of the day, it is a hypothesis, so there's some wiggle room, if you will. Okay. Now, it does have to be based on some sort of information. The main ones or the major ones that we tend to use are things like physical or morphological features or traits and DNA or protein sequences of genes. Okay. So in a tree, we tend to place or locate excuse me, our species at the ends or the tips of the lines, and we call these the tree's branches. Okay, so for example, I'm going to do it sideways, because hopefully that'll give me enough space to write. Okay, that was painful, but anyway, so let's label this A, B, C, D, and E. Okay, so these guys, those are our species of interest. And this here and there, those would be what we call branches. And then here or here are what we call branch points, or sometimes they're called nodes. Okay. And realize in terms of how we're running with the information, ancestors would be on this end. And then these would be our present day species, our modern species. Okay. Let me give you a minute to finish copying that down, and we'll go from there. Okay, so I'm going to redraw this tree and point out a few more features to you. So same situation. But now I want to talk about what's happening at these branching points. Okay, so every time you get to a branch point or a node, we've had a divergence event. We've had something that took a group, one single group of organisms, and caused that single group of organisms to split into two descendant or two different groups from the original. Okay, so that's what's happening at each of these nodes or these branch points. So nodes actually show us the most recent common ancestor for everything that descends from that branch point. So bear with me if you will. We have a branch point here and here. We have another one here and another one here. Let's go through and we're going to label those. Let's call this one 1, 2, 3, and 4. Okay. So different branch points are going to give us different bits of information. So for example, branch point number 3 shows us the most common ancestor or the most recent common ancestor, of species A and species B. Okay. Branch 4, oh my goodness. I hate when it does this. Okay, branch 4 is going to show us the most recent common ancestor for groups C and D. Okay, if we went to branch 2, or branch point 2, not just branch, but branch point or node 2, my bad, we're going to be looking at the most recent common ancestor for A, B, C, and D. Okay, and then of course 1 is going to be the most recent common ancestor of all of the species represented by this tree. Okay. Now, this section, this one line, if you will, right here, this is referred to as the root. Okay. The most recent common ancestor of all the species in a tree is always going to be found at the first branch point above the root. Okay, so again, giving you a few minutes to copy that down. Okay, so something to take into consideration when we're looking at phylogenetic trees and we're thinking about how related two things are to each other. The more related you are, the more recent your common ancestor. So two species are more related if they have a more recent common ancestor than with another species. Okay. On the flip side, two species are less related if they have a less recent common ancestor. So one of the things that, or one of the methods you can use to figure out how related or unrelated or less related, let's say, sorry, you are, is by doing what's called a walk backwards. So you start at whatever species you are trying to figure out, and you walk down the branch until you get to a node. And that kind of pinpoints where the most common ancestor would be. So, for example, let's say we were looking at D and C. Let me change colors really quick. Okay, so D walks this way, and then it walks this way, and there's my common ancestor. B is going to walk this way, and again, walk that way. Okay, so the most recent common ancestor of A and B is going to be at this node that we've already identified, this one right here. But let's say we were looking at the common ancestor, or how related B versus C was. So this guy and this guy. So if we did a walk backwards for B, we would go down, then across, and then down, and across. So note, we'd have to do the same thing on the C end. Go down and across. Notice we have to go further down and across to get to a common ancestor shared by C and B. So this walk backwards just showed us that D and C are more closely related than C and B. Even though they seem to be very close to each other on the tree, it doesn't necessarily change the fact that they are either more or less related to each other. So if you do that walk backwards, if you think, okay, how far back do I have to go to find a node that the two of us share? That's how closely related we are. Okay, so we're going to stop here with the notes part of things for today. Go ahead and pull your FRQ question. Where's this now? Look. It's not looking. Okay. So let's look at this question. This question is from 2015, so it's a great example of what a more recent phylogeny question could look like. And in this question, they've given you this table to read from. So it tells you the amino acid sequence for cytochrome C was determined for 5 different species of vertebrae. The table below shows the number of differences in the sequences between each pair of species. And it gives you the 5, and we're doing a comparison. So College Board loves questions like this where we're doing a side-by-side comparison of one species to another. And sometimes they do numbers like this, and sometimes they do pluses or minuses or a couple of different things. This is not hard once you understand how to read this question. So, excuse me. All of these letters, all of these numbers. Hold on, I need to change my editing feature there. Okay. So all of these numbers represent the differences in amino acid sequences from one species to the next. So if you read through the table, it somewhat makes sense. So E. ferris shouldn't have any differences when compared to itself. So that number is 0. But if you compare E. ferris to D. Polylepis, there are 21 differences between E. ferris and D. Polylepis. There are 11 differences between E. ferris and G. gallus. There are 13 differences between E. ferris and A. foresteri, however you say that. And there is 1 difference between E. ferris and E. africanus. Okay, so that's how we would approach or read this table. So the question says, using the data in the table, create a phylogenetic tree. If they want you to draw a phylogenetic tree for them, they're going to always give you some sort of structure to go based off of. So that's what you're seeing down at the bottom of your page. So create the tree on the template provided to reflect the evolutionary relationships of the organisms. Provide reasoning for the placement on the tree of the species that is least related to the others. Note they don't want you to explain the placement of all 5 organisms, just the one that's not as closely related to everyone else. So if you think about what I just kind of walked over with you, the further away your common ancestor is, or the less recent your common ancestor is, the less related you are. So that's how you're going to decide who ultimately is going to be our outlier. Okay, so it stands to reason that if you have less differences between 2 species, they're probably more related. So look for the smallest number. That would be my suggestion. Not 0, that doesn't count. Look for the smallest number. The smallest number is this 1. This 1 exists between E. africanus and E. ferris, which means that these 2 should be closely related. They should share a close common ancestor. So I would write E. africanus here, and then here I would put E. ferris. Okay, so now we look for the next smallest set of numbers, and we have a 3 right here. That 3 exists between E. foresteri and G. gallus. So that suggests that they too share a fairly common ancestor, or at least a recent common ancestor. Okay, so go ahead and fill that in where you think it goes. I'm going to give you a minute. Do we have enough information? I don't know. Let's look at some more things. So we know that foresteri and gallus have 3 differences between the 2 of them. But I wonder, what is the difference between gallus and foresteri, or gallus and ferris? Well, between gallus and ferris, there's a difference of 11. Between ferris and foresteri, there's a difference of 13. Okay? And between Africanus and Gallus, there's a difference of 10. And between Africanus and forestry, there's a difference of 12. So that at least tells you that in comparison to the other two, there's probably going to have to be more nodes in between them. So that would prompt me to put A forestry here and G Gallus here. Okay. So now let's look at what we have left. We've decided where 4 of them goes. The only one we haven't talked about is Polylepis. If you look at Polylepis, Polylepis has the most differences between itself and Africanus, between itself and forestry, between itself and Gallus. Okay, it also has a fair amount of differences between itself and ferrous. So that tells us that D. Polylepis goes there. So again, when we're thinking of, okay, well, who is our, oh, whoops, nope, okay. So when we're thinking of who is closely related or not closely related, if we did that walk back, we'd have to walk all the way down this node before we found a common ancestor. So that tells me that Polylepis is our least related. Okay, if that didn't tell you, then the fact that it had such large differences between itself and all of the other species should be enough reason why it's not the common ancestor. Or it's not the most related. Okay, so we've done part of part A. The other part says provide a reasoning for the placement. So this is what my reasoning would say if it were me. And we've already addressed what the reasoning is. Okay, so we could say that, hold on, I'm going to switch the color here because it's a little hard to see. Okay, so D. Polylepis is least related because it has the most amino acid sequence differences compared to the other 4 groups. Okay, and that would be enough. That's enough to justify why you put it where you put it. Okay, so now I was giving you time so that you could write stuff down because I think when I exit out of this, all of the writing disappears. Okay, so let's move back and look at Part B. Part B wants you to identify whether morphological data or amino acid sequence data is more likely to accurately represent the true evolutionary relationships among the species and provide a reasoning for your answer. So this is kind of like a trick question. because they've set it up in a way that suggests that there's only one right answer, when in truth and in fact, either of those things could be the right answer. Excuse me. It's just, oh, what did I just do? Okay. It's just about how you justify or how you explain what it is you're thinking. So let's look at this one. So you could say, I believe amino acid sequence data is the better or most likely to represent true evolutionary relationships. And that would be fine. That would get you a point. The other point would come from your ability to back that statement up. So remember, make a claim, give it evidence, and you know, utilize reasoning. So I believe amino acid sequence would be the most accurate. Let's start with that one. If we did amino acid sequences, what could we say? Well, you'd have to think a little bit here. You'd have to think and you'd have to use your vocabulary that you know. There are lots of species that look very similar to each other, maybe even behave similarly to each other, and would lead us to believe that they're related when in truth and in fact they are not related. Some of this happens because of structures that we call analogous structures. Because our environment is similar, we've adapted similar ways of dealing with the same problem. Okay, so this is known as convergent evolution. This would be a perfect example of why amino acid sequences would be more accurate. Okay, so we could say that similar morphological features can exist between two unrelated species. Okay, and if you really wanted to tie it all together in a bow, you could give an example here. Okay, so you could talk about like dolphins and fish because they have a similar body shape and body dimension. They both have fins slash flippers, that kind of thing. You could talk about bats and birds. Both evolved for flight, but they don't actually utilize them in the same way, and they're not super closely related. Okay, so that would be an example of how morphological features would support that amino acid sequences are more accurate. Okay, so we could round this up by saying um, so similar morphological features can exist between two unrelated species due to or because of convergent evolution. Now, let's say you didn't remember the word convergent evolution. Was there something else you could say? Yeah, you could explain what convergent evolution is because they're dealing with similar problems in a similar environment, so they've both adapted to those circumstances. Okay, well, how would we support morphological data? Well, you know how I like to remind you that biology frequently works in opposites? This is where this would come in. So, morphological data could have been our right answer, and our support could be, excuse me, that similar molecular sequences can sometimes produce different morphological features in related species. So, similar DNA sequences can produce different structures in related organisms. And if you wanted an example, you just have to look to our own species, chimps versus humans. Evolutionarily, we are closely related. But when you look at our features, no one's going to mistake a chimp for a human. Okay? We have some similarities for sure, but we don't look exactly the same. Okay, so at this point, the instruction part of this is over. And what I'd like you to work on is the biology basics phylogenetic trees packet. It's 11 questions, 2 trees, and there's some basic information on the front that could be useful. And then when you're done with that, I'd like you to work on the types of selection packet. It's the one with the graphing and the snake. So that's a little bit on a different train of evolution. And then you have a phylogenetic packet over like bioluminescence or something like that. So those are the 3 things you should be working on, or anything that you didn't finish from the class before, or your essays, or your take home test. You have tons to do. Okay, so hope you have a great day and a great weekend. And I will see you guys bright and early on Monday morning. Bye, y'all.